I know after the lunch everybody's a little sleepy, so I try to make things a little more interesting. This is look like a bad title. Is too complicated to understand. Let me pick the keywords. Oh, urinary. I'm very interested in urine. I think urine is probably even a better biomarker source than blood. Let me explain why. So that means a lot of people are looking for biomarker in not the optimal, may not be the right place. I don't know if that's too harsh, but let me explain why. I saw a few of presentations show the definition of biomarkers. Some says it's a protein or a metabolite. Some says it's a state of disease. But I think biomarker is like something that when you are healthy and you change to gradually to disease state, a lot of things changed. Some of the things changed are measurable. Some of them are not. Those measurable things are biomarker. So overall, biomarker is a change, that change from the healthy state to the disease state. So we are actually looking for a change. So that's why when we design a biomarker experiment, we always have at least two groups of samples. One is control, the one is diseased. One healthy, one diseased. Why? We always have to subtract, to compare, to show the difference. That difference is actually a change. So we are looking for a change. Where should we look for change? Are there a lot of changes in blood or more changes somewhere else? If you still remember the physiology textbook, the most important thing in physiology is the homeostasis of internal environment, which means the blood tends to be stable. Our cells are happy in a stable internal environment. We want the blood to be stable. We have all kinds of mechanisms, the liver, the kidney, the skin, the lung, to remove all the unwanted things in the blood to make the blood stable. So most of us are looking for changes in a place where changes are rarely happened, or happen soon and then disappear soon. So looking for biomarkers in blood is like playing hide and seek. You have to find the change before it being removed by liver or kidney. That makes things hard. But in urine, a lot of changes getting to urine. But also there are other changes getting to lung, get to liver, get to skin, but they're all easily to get contaminated by the feces, by the things in your stomach. Only urine. It's so pure, so clean. It's in the bladder for hours, just waiting for you to see what's happening in your body. So I'm interested in looking for biomarkers in urine. Actually, I talked about those last year. That's why I didn't prepare to do that. This year, the organizer asked me to give a workshop on preserving urines economically and easily. I was wondering, is anybody interested in urine at all? So before I do that workshop, I want to do an advertisement just like this. Urine is important for you too. And before, I always say there are more opportunities in urine, in biomarkers in urine. Today, I would also want to emphasize that the risk for not touching urine. I will give more evidences in the workshop, but actually, there are more and more evidence showing that for some of the biomarkers, they perform even better in urine. So you have like opportunities in urine, and you have risk in urine too. If you don't touch it. If somebody tests your biomarker in urine and it performs better, that's a big loss for you. That's why I think it's important to at least take a look at why we should look at urine. So since I talked about those last year, I didn't prepare to talk about that. This year, I would want to say, when we look at the urine in biomarker, the situation is very different from looking for biomarkers in blood. In blood, everything's stable. It's very hard to find a change. But in urine, it's very different. There's so many changes. It's so hard to find the one that associated with your disease. Let me show this. This is actually one of the pictures never get published, because this is the urinary proteins collected from healthy individuals in my laboratory. Graduate students, they're all healthy, young, but the patterns look different. That's probably one of the reasons not many people working on urine. One of the pioneers, Dr. Moses, just gave us presentation yesterday, showed beautiful result many years ago, but not many people working on urine, not following the suit. I was wondering why. I guess a lot of people stop here. When they see there's so many differences in healthy urine, we don't see a smooth baseline. How can we find biomarkers in this kind of environment? But I see it differently. If biomarker is a change, and there's a lot of change, there's a lot of opportunities. That also means they're all healthy, there's changes. That means there are some physiological changes can be reflected in urine. If physiological conditions can be reflected in urine, pathological conditions should be reflected even earlier and more sensitively. So that's why I think urine is a better place to go. But there's a lot of problems in looking for. Today, I want to talk about one problem is when we always have two groups of people, healthy control and patients. We try to stratify with gender, with like same group of male, same number of male here, same number of male there, and age, we all stratified. There's always one problem we can't do it. That's this medication. Ethically, we cannot stop the medication of the patient just for the sake of biomarker discovery. But we can't give same medication to the healthy control either. So when we do the profile of these two groups, there's healthy and disease. At the same time, there's closely associated another factor, which is no medication and medication. That make the balance change. So we have to assume that medication may not change the biomarker. We hope. If the medication do not change the biomarker, the whole scheme looks fine. But if that's true, I think biomarker, the medicine will definitely change the biomarker. Let's think about it. Biomarker is you're from healthy to disease. Medicine is change you from the disease to the healthy. It's supposed to remove some of the biomarkers that disease causes. And at the same time, it probably produce other changes too. So medicine helps the patient to change from disease state to the normal state, and medication must change biomarkers. Not possible. When you are completely healthy, you will see those biomarkers should be gone. That's the effect of medication. So here, not many people study like biomarkers of drugs, of medications. So I wanted to show some of the examples that we did in the laboratory, some of the diuretic, anesthetic, and anticoagulants to see if they really have effects on urine. I try to make it faster in those details. Different kind of diuretics. Basically, it's a self-control. You take the urine and give the animal medicine and see what happened to the urine afterwards. That's the result of diuretics. You see all the rats in the disease in the medicine treated group changed, and some of those proteins that changed were considered biomarker before. That means when they talk about those biomarkers, I don't know if they really noticed that some of the medications may cause that biomarker to change too. That's the same things happen too. And there are some homologs were considered biomarkers too, and they are all easily affected by diuretics. And these two are the anesthetics that we used to test. Similar thing, the similar result. Some of them are considered candidate biomarkers, some of them are not. And those are the validations. I don't want to go too much into the detail. And the third one is actually we tested for if urine is a better biomarker source. We try to change the function of the blood, see if we can see more difference in blood or more difference in urine. One of the fundamental function of the blood is coagulation. Very basic. No coagulation, people die. So when we change with anticoagulant to change the function of blood, guess what will happen more sensitively in blood or in urine? Actually, with the same standard, same approach, everything's the same, we can see more changes in urine and more changes in urine and less change in blood. At my title, I said, in urinary biomarker study, we should take care of the medication, the drugs. But actually, even for people who work on the biomarkers in blood, you should take care about the drug's effect too. Even though there's only small number of proteins change, but they may actually change your result. Let me make an example. Some of the people with the stent in the body, they may use anticoagulant. But when you try to study the effect of stent or the atherosclerosis, you may not notice all the people with stent, they may have some kind of anticoagulant. And those healthy control, you can never give them anticoagulant. So that marker may come from two sources. One is drug, one is the real effect. So one way is we study the drug, the effect of drug in animal, and when we study the biomarker, we can subtract that one. So I want to skip that summary too. This is another important thing that I think is very useful for studying urinary biomarkers. Just like I said, for studying biomarkers in blood, the problem is to find change. There's so many proteins, and the body only allows little change. You cannot have too much change because people will die if you have too much change in blood. But in urine, you have less protein. You can tolerate a lot of changes. The problem is to find changes. It is to find association, the change and the disease association. How to do it? For clinical studies, it's very hard to control. And what we eat, what we drink, if we do exercise or not, they all change urinary proteins. It's very hard to have exactly two groups of people, diseased and healthy people, exactly the same. Another reason is medication. It's very hard. But I think we can minimize the factors by using animal models. So if we find the clue from the animal models and then validate in human sample, maybe it's much easier to find biomarker this way. That's actually that I prepared. So I've talked about earlier in the talk. Biomarker is a change. Urine accumulate a lot of change is a better biomarker source. That's why we want to save it. save it easily, economically, at room temperature for a long time. So I want to just leave a little time for questions.